All right, I think we can get it started. Um, so next week will be the first exam, and I hope you are all aware of it. And it will be a closed book exam, which is lucky you, and because the first time when I gave an open book exam, and actually I designed the exam like 3 hours. Nobody finished in 3.5 hours, and we have to extend that to 4 hours. But now it's a closed book. It's much easier, and you have different type of questions, and that there are multiple choice questions, and there are a few questions like you have one single choice and the correct ones. There are some questions that has multiple correct answers, and I will not tell you which one is correct, how many correct answers are, but you will see the description. And there is a few questions about the essay type of question. So that will be, it's going to be quick. So if you know it, you write it. If you don't know it, you don't have time to study it anyways. So it should be fine. Any questions so far regarding the exam? Yes. Might, I don't know. I haven't made any questions. That's why that I'm not going to constrain myself to make those questions. But bottom line is you are not required to be a math major to answer any of these questions. So if you're a biology major student, then you should be do just as fine as the people who has a math major, who has a math background. So don't worry about that. Any other questions? It may be 10 single choice, and maybe 5 multiple choice questions, and maybe 5 or 6 essay questions. So I don't know. So we'll see how much I run out of gas and cannot make more questions out of this. Other questions? Yeah. There will be straightforward questions. There will be tricky questions. Yes. It'll be very tricky. Are happy now? No, yeah. But the bottom line is that what I want you to understand is the concepts, the basic concepts. Okay, but you will see. So don't over worry about that. And also that we will use the first 2/3 of today's lecture talking about the stuff I want to talk about, and then the last hour will be Sean Courtney will talk about the hands-on for the RNA sequence for the DNA sequencing part, and he is going to give you some homework, and it's not due next week. It is due the week after, so you have one more week after the exam to do that. Okay. After that, I'm done, and from RNA sequencing, that will be Zhang, and for 3 more lectures, and then 2 more lectures for Dr. Jun Wan on the ChIP-seq and ATAC-seq. All right, and that will be the first time they teach that, so I'm sure you guys will do a good job. All right, so okay, let's get started for today's lecture. And actually, we still have a lot of things to cover for the DNA sequencing part. And the first part of it, I'm going to talk about the variant prioritization and rare variant association studies. And the last part will be on the metagenomics, just very basic concept. Okay. Again, that a little bit review on what we discussed in the previous lectures. So for the DNA sequencing, there are several computational bioinformatics related questions. The first one is the mapability, because we know that the human genome is repetitive. Actually, 52% of the human genome are in the repetitive regions. And the non-unique mapping can cause analysis errors, or in some places, they simply cannot be aligned. So you can see from this figure that if your read length is about 50 base, for example, for human genome, it's about 80% of the region is alignable. And other regions, you simply, it's too repetitive. You cannot using 50 nucleotide base to uniquely align that. So, and mapability is region specific, and sometimes it's algorithm specific as well. And we spend quite a lot of effort talking about refined alignment. And those are focusing on the regions that has small insertion deletions, like something like this. And so presence of indels is small insertion deletions, will cause local misalignment. And the reason that for that is the initial alignment is minimizing the matches, mismatches for one single read. Well, for the refined alignment is minimize the mismatches for multiple reads. So if we need to do the refined alignment, actually there are two steps. The first one is to identify suspicious regions. There are several rules, and the second part is going to be conducted the refined alignment. So basic that will include three major steps. The first one is enumerate potential haplotype candidates, and then conduct gapless alignment on all the haplotypes, and then further calculate the likelihood by including new haplotype, whether that will increase the overall accuracy. So that is for the refined alignment. And this figure shows you that one of the results from their original papers, you can see in this region, before do the refined alignment, there are multiple SNPs being identified. At the same time, there are a few reads that supporting there is an insertion deletions. In this case, it will be deletion. And after refined alignment, there's actually the deletion, but there's no related, thank you, reads that mismatches that is involved. So that is for the refined alignment. This is very, very important concept that we really need to think it through. And the second part that we talk about, the third part actually, is the base quality recalibration. The question we want to address here is when sequencers give you the ACGT reporting, actually for every single letter, they will report a quality, which is, if it's 20, that means you have 1% of chance make mistake. If it's 30, that means 0.1% of chance. But whether that reported quality is accurate or not, and from the analysis, by looking at what they report and the so-called empirical analysis, those purple dots, we know that all of the major manufacturers, when they report the quality, is not very accurate. So basically, these are sometimes they actually what they're reporting are a little bit more overly optimistic. So it's if it's 30, actually it's 27 or something like that. So that will give you a false confidence. And we know there are many different parameters or features that is related to the misreporting. That include the reported qualities, positions inside the reads, and sequence contacts with nucleotides, and many other things. So the GATK package do give you the option to do the refined base quality alignment. And then you see that after this recalibration, and after this recalibration, that the reported quality is pretty much similar to the empirical qualities. So those misreporting has been corrected. And then we spend some effort talking about the variant identification part. And so basically the general idea is we spend most of the time talking about it. If we look at identification for the diploid genome, basically using a Bayesian-based approach, if you still remember that. And so in this case, you can see there's a G and there's an A here, and reference genome is A. Probably in this case, this will be a homozygous variance with this particular A as a mismatch or sequencing error or something like that. So this is for the diploid genome. However, this conclusion is really depends on the study design. if we are not talking about diploid genome, meaning that human genome or germlines, so if we are talking about cancer genome, if you observe this particular pattern, that simply means only a small proportion of the cells have this A allele and larger is a G allele, and that is a somatic mutation. If this is a pooled sequencing, meaning this is the data being identified from a pool of multiple individuals, this will tell you a small percentage of the individuals that in this pool have A, others are G. If this is the RNA sequencing, that might mean this is a homozygous variance. Actually, one allele is G, one allele is A. However, A allele doesn't express too much, while G allele expresses in a very high level. So this is an allele-specific expression. And also that towards the end, we also discussed how are we going to potentially increase our statistical power to identify genetic variants by borrowing the information from multiple individuals joint calling. So one specific example was in the 1,000 genomes when they sequenced only 4x coverage by average, but they were able to borrow the information from other samples to increase the statistical power. And sometimes they can also borrow from their own genome in other related locations. So that kind of you have increased the confidence. So those are the things that I'm not trying to force you to learn the math. It's very difficult. It's kind of haunting to many people, including myself. But I want you to understand the concept. What are the information they have borrowed and why that could increase the statistical power. So this is the variant identification part. And towards the end, we also talking about the variant quality recalibration. For this part, the question they want to address here is when we identify a variant, when we give a quality score for that particular variant, at the same time, we have many other sequencing features related to this variant, like how many reads I observed, what is the sequencing depth, what are the mapping qualities in this region? Is this a uniquely mappable region or not? Is that there is a strand specific or not? So in the old days, before we have this GATK tool, what we use is we set up a lot of different arbitrary cutoff for every single parameters. Sometimes the data will become really not reproducible anymore because my group probably is doing slightly different from the other group. It's just simply because the parameter setting has an issue. So for this one, what they are trying to do is let's summarize all these features and give a unified score to everything. So the general idea is they will using a machine learning based approach like this, but they generally derive a rule from a set of very highly confident known variant locations being identified. So these are the ones that I have a lot of confidence to say they are right. Okay, let's see what they have been doing and what are the feature distributions. And then when I have another variant I have never seen before, and if this distribution is very similar to the known ones, and I will have confidence say they are right, otherwise they might not be. So basically derived a computational model and train a computational machine, and then for all the other variants, let this machine to decide whether they can be a correct variant or not. Again, the computational part of the formulas are very, very difficult to understand, but this is the general concept they're trying to get through. And we also spend a significant effort talking about the structure variations. I'm going to use this one. So if you look at these slides, there are many different types of large structure variations. These are not small insertion deletions, rather they are large ones. But the general information they are using are these three types, which is written here. Read pair, so usually we do this pair and read, and by looking at the distances and then the relative locations, we are kind of can identify the structure variation. The read DAX, and this is more for the copy number variation and deletions. And split race, meaning that one sequence race, and then somehow the variant occurred in the middle. And so different algorithms, there are many different algorithms. They basically using these three type of information. Of course, that there will be assembly-based approach by using all these three type of information to identify structure variations. And I have listed a bunch of algorithms that you can select and when you do the real application, but in general, they're using these information. So when we talk about all these things, you may not notice that I'm not specifically talking about one single algorithm. Rather, I try to summarize different algorithms, tell you what are the information being pushed into it. So in this case, in the future, if a new tool comes out, and then you read the papers that will go into your knowledge base, are they using this information better or that information better? Or they're using some new information. So that will increase your knowledge base in the future. And that also make my life easier. So next year, I don't have to redo all my slides because new tool comes out. So this is the summary slides. So you can see that the major components in the DNA sequencing analysis, we start from raw data and then we do the QC and then do the alignment, refine the alignment, variant calling, variant quality recalibration. We are pretty much at it here now. So what we try to introduce today, the idea is after you identified a few thousand, a few hundred, a few million, doesn't really matter, SNPs, and how are we going to further make the biological stories? What are the tools that people often use? So this will come to the major topic of the variant prioritization part. So the first part is let's just have a list of what are the information being used. And I think I introduced this slide in a few lectures ago. So the first group of information people often use is the genomic loci. So meaning whether this, where these variants are. Are they in the intron, exon, junction, UTR, non-coding regions, promoter enhancer regions? So what are their relative genomic locations? And the second one is to look at the population-based analysis. So the database people often use include the dbSNP, Southern Genomes, Seattle SNPs. There are some new ones coming out as well. And then we want to look at their molecular functions, whether they are changing the amino protein structures or not. If they do, they probably is non-synonymous SNPs, meaning that they will change the amino acid sequence. So many of them are related to splicing, whether they're on the splicing site or in the exon or in some other regions. And then whether they are in the promoter regulatory regions. So for this part is really separate different SNPs based on their potential molecular functions. Okay, so the next few slides, we're going to introduce what are the general information people use for this individual level of annotations. Okay, so the first one is annotation based on the genomic loci. As I mentioned earlier, this is based on known gene structures, which part is exon, intron, UTR, microRNA site, you name it. And those are usually exist in the database format. And it's either in the RefSeq or UCSC gene ensemble gene code. So those are different levels of gene annotation that tells you where is the gene, where is the promoter, where is the enhancer regions. And actually, most of these resources are also in the UCSC genome browser. If you are not very good at programming, you can directly go there to check and whether there is where this particular SNP is. And then speaking of genomic loci, people also very much interested in looking at evolution information, meaning that whether this is conserved across at least the vertebrate animals. So the general idea is if this SNP or variant locates in a conserved region, they are more likely to be biologically useful and therefore more likely to generate a pathological impact. And then they also sometimes rely on other type of functional annotations. For example, transcripts are expressed in the organ or tissue most relevant to the disease. The reason for this is, for example, if you study a liver disease, and then you find out the SNP. Well, you find out the SNP, if that gene doesn't express in liver at all, it's probably not important in the disease you are studying. But this, we need to be really careful about this. This is some of the hypotheses you have to chip in, assumptions you have to chip in your analysis. And then every time you do that, you will have the benefit of potentially increase your power, but at the same time, if your assumption is not holding, and probably your conclusion will be wrong. So sometimes this can be very difficult because you don't know exactly where to look at, right? For example, if I study hypertension, should I look at the endothelial cells, the heart muscles? So what I'm going to look at? Sometimes it's that we just don't know. And in other cases, it can be misleading. Just because it's not expressed in the normal tissue doesn't mean it doesn't express in the disease organs. So just to be a little careful on this. But this is, nevertheless, this can be a very important level of information people can use. And then not only that, you can look at the promoters, enhancers, and DNA hypersensitivity regions, whether these SNPs are in those regions. And most of these regions we can download or find out from the ENCODE project or other gene annotation level projects. And all of these informations almost are documented in the UCSC genome browser. You can directly go there to check. So the second level of annotation is going to be annotated based on the populations. And you can see that the first important one is going to be dbSNP. It is a database that is hosted by NCBI and include more than SNPs actually, although it's called dbSNP, actually it has indels and microsatellites and other type of genetic variations. Actually, they not only document the human variants, they can be animals as well. But in most of cases, we probably only focus on human studies. And it also includes many different levels of databases, like 1,000 genomes sequencing data, the variants that has already been documented or included into the dbSNP database. And we also mentioned previously multiple times about 1,000 Genomes project. It turns out this is something very useful to do the annotation. And keeping in mind, when we talk about a 1,000 Genomes project, those individuals, when they were recruited, they have not shown pathological phenotype, meaning they are healthy individuals. So the reason that we use that a lot is if you are studying a rare disease, especially early onset disease, and if you identify a variant, if this variant occur pretty frequent in a healthy population, the chances they are not relevant to your disease, right? So that's why that when people do the annotation, they will go back to the 1,000 genomes data to see whether I have seen these variants before, if I have, and what are the allele frequency of those. But we need to be really, really careful when using it. This is a strong assumption. It usually works really well for the early onset disease. But if we are studying, let's say you study a childhood cancer, probably okay. But if you are studying like a hypertension or Alzheimer's disease or Parkinson's disease, those very late onset disease, just because 1,000 genomes individuals, they didn't show that, then potentially means they have not been old enough to experience those disease yet. So just be very careful when you use this assumption. But anyway, so annotation based on the population, 1,000 genomes is a very, very important database. So if you use the ANNOVAR, for example, to do the annotation, they will also tell you what are the allele frequency of individual SNPs, but you also have the option to report for each individual of the 5 subpopulations, what are the allele frequencies within this population. And there's another database that recently become really popular. It's called Exacta. It's called the Exome Aggregation Consortium. And actually, these are only focused on the exome regions, but they now have over 60,000 unrelated individuals. They are not necessary to be all healthy, but they are, that's the important level of data that is out there. And so when we talk about disease database, if you find a variant, and the chance is the first thing you want to see, okay, have I seen this variant before in certain disease? And that will come to a known database like a ClinVar database is probably one of the most complete. I wouldn't say complete, but at least publicly accessible data sets. So using this is completely free, and they are open to the public. And it's also maintained by the NCBI. It's an archive of assertions of clinical significance for variants related to the phenotypes, and public and freely accessible, and submission-driven database and curation supported by NCBI staff. So you can go there, that database to look at. Basically, most of the records in the ClinVar database are reported to the NCBI database by the clinical laboratories. So they will have to confirm that certain variants is being observed in this particular disease, and they can start to report into it. And they also, if you go deeper, they also have different levels of information. Like some of the variants only reported by one laboratory, they will give a level of confidence. Some of them are being reported by more than one. Others are sometimes even being validated. So there are different levels and give you a different level of confidence. Some of them actually can have conflicting records, and they also tell you that. Okay, so it's very, very useful. So we talk about the annotation based on the genomic locations and the populations. And the next important thing is to annotate the variants based on the molecular functions. So we know that a SNP or small insertion deletions can cause this potential disruption on the protein functions. Like if it's nonsense SNP that is within an exon and translated amino acid change to stop codon. So basically you'll end up with a shorter protein. If this occur very early, that can trigger the nonsense mediated decay. Frame shifting, that is more for the insertion deletions, and the non-synonymous variants, splice 5 or 3, and those are the variants that is on the splicing junction sites that is very much likely to impact the splicing regulation. And in the coding region, but it's synonymous mutations that doesn't change amino acid. It's in the 5' UTR or 3' UTR regions. It can impact the microRNA binding sites, or in 5' UTR regions, it can impact the translational control. And it's a near gene that is usually in the promoter regions, and the intron regions that is between the exons. So those are for different molecular functions, and people also can start to do different level of annotations. So the first thing is a lot of people, actually most of the people focus their efforts to look at only for the non-synonymous variants. And the importance of this is that they do cause amino acid substitutions and therefore potentially affect the phenotype of the host organism. And it constitutes more than 50% of mutations known to be involved in the human disease based on some of the statistics. So we know they are kind of pathogenic. Many of them are pathogenic. There are a lot of them being documented in the human gene mutation database or OMIM database already. And we know that whole human population has many non-synonymous mutations, and they have a lower minor allele frequency distribution than those that are predicted to be functionally neutral. So this is an important concept, actually. So normally the disease-causing mutations, they tend to have lower minor allele frequency in the general population. The reason is very simple. It's evolution selection. And because if they disrupt our genome, make us sick, and we'll have less likelihood to produce, so that they are not going travel with our genomes. So that is a very important concept. So if I have an exam question, for example, asking you, is a lower allele minor frequency means more pathogenic or something like that, and you probably have an answer for that now, right? So yes. I'm not familiar with the disease you're talking about, but in general, that is the general trend. So probably for certain disease, it's not going to hold, and also that again, this is also coming to whether it's a disease that is impact the offspring, the reproduce or not. It's before that that the next generation is being generated. So if it's Alzheimer's disease, probably they are not being have any impact or relationship with the allele frequency. So it's a disease specific, but just to keep in mind, in general, that's a general rule. So, and that is also based on different levels of overall statistics. But you're right, for some certain instances or diseases, they could be more advantageous. So the general strategies for non-synonymous SNP functional prediction, and so we can see that disease-causing mutations are more likely to occur at positions that are conserved throughout evolution. We know that. So this is a figure, and probably you cannot see from very back, but this really tells you that the red ones are the disease-causing ones. So you can see that they tends to be in a much higher conserved regions. And another one is the relative probability of disease-causing mutation is the highest in the protein interior and the lowest on the protein surface. So this is another important observation that people made is when you look at a protein structure, and if the mutation occurred inside of the protein structure, they tends to be important, causing disease. If it's outside, and they tends to be less risky SNPs. The reason for that you can think of is that there's a structure, and if the mutation inside causes amino acid to become bigger or smaller, or hydrophobic or hydrophilic, or the charge changed, and then the entire protein structure will be altered. But if it's outside, and it may only impact the local region. And this is a similar thing. It's not a generic rule. So in many cases, important SNPs are on outside. It's sometimes when they are in the protein-protein interaction regions, they can be in the outside, but the SNPs over there are very important as well. So, but this is for generic observation, so that we can see that the disease-causing mutations tends to be evolutionarily more conserved at the same time, so that they tends to be inside of the protein region. Actually, we have a paper about this, and the outside ones, the protein-protein interaction part. But I'm not going to go through all this flow chart. What I want you to understand is there are many algorithms people use to predict whether this non-synonymous SNP can change the protein structure or not. Okay, so the information they use is sequence information, structure information, and annotation information. So those are the information all different algorithms use. For example, when we talk about SIFT, polyphony, those are the tools that you can directly plug your SNPs in, and they will help you to predict whether they're pathologically important or not. So we see that there are actually many, many such algorithms. There are over 2,000 of them back in even 10 years ago. And the most famous ones probably is the SIFT and the polyfin, not polyfin, it's polyfin 2. And, but I want you to understand that is SIFT actually only use the sequence information, while polyfin use both the sequence and the structure information. Okay, but in general, all the tools are using this information, but in a different way. So which algorithm to use? That can be a very difficult question to answer. And direct comparison among different algorithms are very difficult. And because every single one of them will claim they're doing better than others because they are based on the assumptions they established when they do the testing side, and they are going to, I mean, either intentionally or unintentionally pick the ones that shows they perform a little better. But in general, when we look at the structure-based information, is if you think about the SIFT, is using sequence. Polyfin is using sequence and the structure. You may think, of course, I'm going to use polyfin because it's using more information. But actually, if you're based on the structure-based information, there are potential problems as well. First of all, you will have less prediction because not every single protein we have a structure yet. And the training data set, we don't have a structure either. And sometimes it can be misleading because the structures are usually generated in the tube. It's not in the system. In the system, they may have other proteins binding, and I mean, everything could change. Using sequence-based information, basically people use the homology and the ortholog in different species, and they think they will potentially represent the same function, but in some cases, they may represent different functions. So just be careful about using which algorithm. But by the end of the day, you do not have a lot of choices. You just end up using all of them. And then you try to see, okay, which one is being predicted by most of the algorithms I tried. So they potentially are more accurate. But actually, there is a few studies that reported a new trend by using ensemble-based approach. So meaning that, for example, this CAD, basically what they did is they tried multiple ones. And then they have a training data set. They use this and combine that using machine learning model to give you one unified score rather than 20 scores from 20 different predictors. So that is sometimes can give you a pretty good result. So people start to tend to use the CADD, which is this ensemble-based scoring system to prioritize different SNPs. So those are the ones for non-synonymous SNPs, meaning they will change amino acid and therefore change the protein function. But what about the synonymous variants? So they are traditionally called silent mutations and synonymous and may cause, so they don't cause the changes in the protein sequence. So most people don't think it's important, but actually they may have impact on the protein expression levels, conformations, and different level of functions. So this is a pretty early review paper. It tells you that many different organ-related diseases are actually related to splicing, RNA structure, rate of translation, and they are related to the synonymous SNP, the SNP that doesn't change amino acid sequences. Okay, so they were considered as no effect on the fitness of organ and would be neutral during the evolution. Okay, but if we look at a few publications, and this is the one that you see the title of this article is non-synonymous and synonymous coding SNPs show similar likelihood and effect size of a human disease association. That really tells you that there is not too much difference between the synonymous and non-synonymous SNPs. So they got this conclusion based on survey over 2,100 different GWAS studies over 21,000 SNP cases. And so you can see that this is the left one is the likelihood of disease association, right panel is the effect size, and these two arrows representing the non-synonymous and synonymous SNPs. You can see there's really very, very little differences, okay? And the same conclusion holds for the cancer studies as well. So you look at this title of this paper is synonymous mutations frequently act as driver mutations in human cancers. I'm not going to go to the data slides, but you can see that synonymous SNPs can be very, very important as well. Why is that? So if they do not change the amino acid sequence, how come they can be biologically important? So one of the mechanisms is shown here that is impacting the splicing regulation. So meaning that if this is a very simplified cartoon, right? So this gene has 3 exons. There are 3 SR protein binds here. So you can see that the mRNA spliced outcome look like this. But if there's a mutation here, that this mutation causes the middle exon, the SR protein is not binding there, the splicing pattern may occur from here and here. So you are having a 2 exon SNP gene. So this one SNP, if you look at, they probably don't change amino acid sequence. However, they're causing the entire domain to be missing, okay? So that is one mechanism that SNP can cause the splicing changes. The second one is it can cause the global stability and the local stability of the RNA molecule. So the general idea is you can see that we know that mRNA form these secondary structures. So this is one structure, and the right part is a different structure. The difference is here. So there is like a bow here. And this could be because of the SNP that causing that pairing did not happen. So we know that this one has a much higher energy than this structure, meaning this is less stable, okay? And this is another example, but it's a reverse. So this is, you can consider this like a transmembrane part, but this one is more stable in here, and this one is less stable as well. So you can see that this one nucleotide difference, a SNP, can change the RNA secondary structure, therefore causing the stability of the RNA molecule change. So that's another potential strategy. I mean, the mechanism. And the third one here is genome-wide trends in the codon usage. So what that really means is we know we have 3 nucleotides become an amino acid, right? So tRNA is facilitating this process. But we have, I mean, if 4 nucleotide letters, if every 3 letters become an amino acid, we should have 64 different options. But we only have 20 amino acid sequences, amino acid choices. So that means there are redundancy, right? So if you have a SNP, did not change the amino acid. However, that will cause, this SNP will cause the usage of a different tRNA. So the 2 different tRNAs, they may have different availability, meaning that one tRNA is much more than the other one, okay? In that case, when you do the translation, you need to wait until the tRNA comes. So if the tRNA, for example, is not available there, that will cause your translational rate to become much slower. So all those different mechanisms, this is something called codon usage. They can cause a different outcome on the proteins, on the protein level, either structure or expression levels. So that can be different. So the molecular mechanism for synonymous SNPs that can impact the post-transcription regulation, including splicing site, splicing regulation, sometimes microRNA regulation, and then for the it can impact translational control, and also it can also impact the protein stability. So there are different methods to try to predict the SNP, especially the synonymous SNP, how they are going to impact the splicing outcomes. So And there are very simplified annotation-based studies. So if your SNP occurred at this splicing junction sites, and that is being considered as automatic will change the splicing outcome. Is that true or not? May not be perfectly correct, but that is kind of the generic rule. And those SNPs will affect the formation of the splicing. And also there are many regulatory regions for the alternative splicing, including the ESS and ESE and the ISS and ISE. Those stands for exonic splicing enhancer and silencers, and intron splicing enhancer and silencers. So if in those regions there are SNPs, that will impact the enhancement of the splicing or inhibition of the splicing. So those are very important cis-action regulations. So to look at this information, there are multiple databases looking for this splicing regulation cis-acting components. And in 2015, there is a paper published in Science. So basically, they use a deep learning method. So basically, they look at many RNA sequencing data, and then they were trying to derive certain route, eventually develop a machine learning tool that you give the SNP, they will tell you whether this SNP is going to impact the splicing outcome or not. So that is being published. It's called Spinner and being published in Science in 2015. So, and if you have a SNP, it's synonymous. You can load your data into their database to see whether they're going to change the splicing outcome or not. And this is something that we developed. We published earlier this year is actually the SNP change the splicing, but we add in one additional layer of information that we also evaluate whether this change of the splicing outcome will impact the protein function. Because only just the splicing outcome change doesn't mean the protein function being altered dramatically. So basically what we did is we use a machine learning based approach. We have the training data set, we have a positive and negative. Positive meaning we know these SNPs are causing the disease, and negative we know they are not. But we look at what are the differences between these two groups of SNPs. Why some of them will cause the disease, others are not. So we look at many different features, including the splicing regulation features. But more importantly, we also include the protein structure related features that is affiliated to the exon being impacted. So by doing this, we develop a machine learning model and change that into a tool. So if you have a new list of candidate SNP, and we can really use that to predict whether this SNP is going to cause an amino acid, causing the phenotype by changing the splicing outcome. So if you look at our result here is the blue line is Spinner results actually. The red one is our predictors. And it shows in both two cases on splicing side variants and off splicing side variants, we see a much higher accuracy in terms of the prediction. But you just feel free to look at it. I'm not going to talk too much detail. This is not my research seminar, right? Talk too much about detail about the algorithm itself. And another level is the variance in the promoter and the 3' UTR regions. So you can see that in the 3' UTR region, they can pretty much change the target site of microRNAs and therefore causing the stability changes of the gene. And they are usually on the seed site of the microRNA. But this requires you to know a little bit more about the microRNA regulation mechanism. If it is in promoter regions, they're usually considered as where the transcription factor binds. So it's something like this, that if in one condition, this transcription factor binds here and causing gene expression by how much, but if the SNP occurred, this transcription factor may not be able to bind there, therefore the expression level will be different. So that is a classical or simplified model of how the cis-actin SNPs is going to impact the gene expression. Most methods identify SNPs reside in all known and predicted transcription factor binding sites. So what we are observing is, and others are using more features like the distance to the, so this is talking about what are the tools to be used to predict the functions of the SNPs in the promoter regions. And we know that transcription factors, their binding are degenerate, okay? And I will make sure that when Jun talk about his part, and he will have some slides that shows this theory in terms of transcriptional regulation mechanism. But the general idea is transcription factor doesn't only bind on GGAA, GAG, or something like that. Instead, some of the positions, they are more important than others. For example, if this position, if G is being targeted or changed, and that will dramatically impact whether transcription factor will bind or not. But if this position is being altered, and they may not have too much influence. So those information is called TF binding degeneracy. It needs to be considered when you evaluate those SNPs. So I'm not going to go detail. This is an earlier paper we published. Actually, if you look at the results, is our algorithm actually predicted that these are the important sites. But you can see, go back to the sequencing logo, they are all the big letters, meaning they are not replaceable. Okay. So those are the SNPs in the promoter regions. Now a few slides talking about the functions of the small insertion deletions. Okay. Just refresh your memory. What are the small insertion deletions? SNPs are one nucleotide substitution, but insertion deletions are you insert, you add a few more or delete a few more nucleotides from your reference genome. So there are two types. One is called non-frame shifted insertion deletions. So meaning that if your number of nucleotides is a multiple of 3, okay, and your end result is going to be increase or inserted or deleted 1 or 2 or 3 amino acid if it's a multiple of 3. And there is another type which is a frame shift. So that means if you plug in 1 nucleotide, you think it's less important than 3. No, because that will change all the codon afterwards, all the codon sequences afterwards. So meaning that every single amino acid after this location will be altered. That is a really big change. So there are frame shifts or non-frame shift in that. And when we start to work on this area, there is really no algorithm or design to look at the impact of small insertion deletions. So I'm going to show you some of the analysis that we did. We found that the red ones are disease-causing indels, and the blue ones are neutral indels. So if we look at the DNA conservation, you can see that disease-causing indels tends to be in the conserved regions. We know it, right? And then we look at another feature, which is ASA. This is called accessible surface area. So what that is, it's also is interior of the protein or outside of the protein. If the ASA accessible surface area is small, that means that it is buried inside the protein. And otherwise it's kind of exposed. So we can see very, very clear distinction that disease-causing ones tends to be buried inside the proteins, and while the blue ones or neutral ones are outside. And then we look at whether they are in the structured regions or disordered regions. Disease-causing ones tends to be in structured regions, and neutral or tends to be in the disordered regions. So by combining all these together, we can build a very, very good predictor, like the AUC is close to 0.9, and it's very, very high accuracy in terms of prediction. So these are the papers actually we published in this area. We published 2 papers on one on the non-frame shift index, the other one on the frame shift index. And then there's another one that we published to see how those small insertion deletions in the exon regions are impacting the splicing. So, but bottom line is this is still an active research area. I think we contribute to the literature in this area as well. So when we talk about different tools, so those are the theories. You say, okay, those are useful, but how I'm going to use that information. And actually, the good news is there are many tools that can help you do that. Most notable one will be the NOR. And this is a tool that you can directly apply it. If you have a VCF file generated, and they will help you to annotate. You need to select which annotation you want them to give you. So for gene-based, region-based, filter-based annotation, they all provide. So everything I talked about today, it actually have the database. They pre-calculated. You just put your VCF file inside, and they will give you the annotation on that. And if you don't like that, and you can use this VEP, which is maintained by Europe, and they kind of they have a web-based server. You load your VCF file into it. It will help you do annotation as well. And also, many popular tools have their own wrappers, like SIFT, Polyfin, CADD. They all have their own website. You can load your stuff into it and to make the calculation. There are some other customized approaches. Summarizing of this, what I talk about today is when we do the variant prioritization, if we look at the whole exome sequencing, generally we're looking at about 30 to 40,000 SNPs being identified. And then we will look at the association studies to see in general statistics whether it occurs more often in the disease population or less often. So that will include the association-based studies or maybe some other studies like family-based linkage analysis. And then we need to look at annotation based on the populations, look at in the 1,000 genomes database whether we have seen these variants before, and then filter based on the annotations, and then do the predictions on their potential functionality. So now going back to this, we pretty much covered this part. So I'm basically telling you a lot of theory, but if you really want to use it, it's really pretty simple. You just use the NOR, it will help you to do the annotation, okay? Anybody knows a little bit Linux should be able to do that, okay? So let's take a 5 minutes break and then we'll talk about the statistical analysis on rare variants.